Welcome to the third part of our video series on photosynthesis. Last time we talked about how light is absorbed by pigments, but those pigments don't exist in isolation in the thylakoid membranes of the chloroplast. They work in collaboration with proteins called photosystems. And so the collection of pigments, other molecules, and proteins that does the light harvesting in the chloroplast are called photosystems. So here's an isolated photosystem, and you've got to put this in the context of the whole thylakoid membrane. So those membranes in the thylakoids are completely covered in these photosystems, and so these span the membrane of the thylakoid, and they are made from numerous proteins, all interacting and working together, holding pigments and doing a lot of complicated chemistry. So what we're going to look at to start with is the basic structure of the photosystem, and then we will look at the mechanism by which light energy is absorbed and effectively converted into a form of chemical energy in the form of excited electrons. So here's our photosystem, and the kind of white or the gray structures, those represent proteins, and these are just models basically showing the general structure of all the proteins involved here. And if you look down on this structure, it looks a bit like a donut with these little kind of alien legs hanging off the bottom. And we're looking at a bit of a kind of slice, an off-center slice. If you imagine you've got your donut and you kind of sliced it like just off the edge here, we're looking inside the photosystem from kind of the side view. So the photosystem has this ring of proteins called the light harvesting complex, as a set of protein interactions which create the reaction center. You can see we've got a number of chlorophyll pigment molecules here. And then there's a couple of types of molecules we've not talked about. The molecule here is called P680, and then there's a molecule called the primary electron acceptor. And these are modified forms of pigments, somewhat related to, but somewhat different from the chlorophylls that you saw in the previous video. So embedded in these proteins in the light harvesting complex are chlorophyll molecules, chlorophyll A, chlorophyll B, and carotenoids. And we're only going to focus on the chlorophylls, so we won't worry about the carotenoids, but we're going to see, you will see a similar set of reactions for the carotenoids. So what's going to happen here? Well, the first thing that happens is a photon of light is going to strike the photon of light will strike a pigment molecule. So here's a pigment here, and it's being struck by a photon of light. So that photon of light hits the pigment, and then last time we talked about how electrons can go from a ground energy state into a higher energy state if they absorb a photon of a photon of light. So an electron in that pigment goes from low energy to a high energy state. But that is really not stable at all. And so what very quickly happens is that electron that's energized in our pigment comes back down. It just drops spontaneously back down to the ground state. So what happens is a photon of light comes in, the electron gets excited, and then it drops straight back down again. So that's all happening in this first pigment molecule that was struck by a photon of light. So photon of light strikes the pigment, electron gets excited, drops straight back down again. So that happens in one pigment molecule. Now when the electron drops back down again, there's a release of energy. So we put energy in to excite the electron, and then as it drops back down on its own, it releases energy, and that's a photon of light. And so that photon of light then strikes a chlorophyll pigment which is adjacent to the first one that absorbed that first photon. And the same thing happens. We get an electron excited, then it drops back down again, and another photon of light is released and absorbed by the next chlorophyll. Now, this is showing you a particular order, but this depends on the arrangement of chlorophyll molecules. And so this photon of light has come in, excited an electron, the electron drops down, and another photon of light is emitted and captured by a neighboring chlorophyll. So this is called fluorescence resonance energy transfer. And so the electrons are getting excited and then dropping back down, and when they drop down they emit light. So eventually an electron in one of these pigments, which is close to the reaction center, remember the reaction center is kind of down here, a pigment kind of close down to the reaction center at the bottom as we're looking at it, an electron there gets excited, drops back down, the photon of light is excited. And so that photon of light, if it's just the right wavelength, when it strikes this molecule called P680, the same thing happens. An electron gets excited, then it drops back down again. But there's no dropping back down again. And so the electron gets excited and it gets held. And that little asterisk there indicates that the P680's got an excited electron. Now, in all of these pigment molecules that we talked about to start with, the electron went up and came straight back down again. Here in this P680, this special chlorophyll as it's called, when it absorbs a wavelength of light at 680 nanometers, the electron goes up and it's held very briefly at that point. And so once it's held at that point, this molecule called the primary electron acceptor, this molecule here, the primary electron acceptor, can grab the electron from P680. So P680 loses an electron. Now think about it. P680 loses an electron, and so loss of electrons, that's oxidation, right? And so P680 is becoming oxidized. It's lost an electron, and that makes it positively charged. And so we show that with this little plus symbol here now. So now we know that P680 is being oxidized, and the electrons have been passed to the primary electron acceptor. So the primary electron acceptor has gained that electron. We can see that from the little negative sign there. And so the primary electron acceptor has been reduced. And that's very stable. So we've now got a very highly energized electron. But we've got a problem. We've lost an electron from down here. Now we can't just keep taking electrons from P680. They've got to be replaced. That would just, wouldn't work if we just kept taking electrons from P680. So where do those electrons come from? That's where water is going to come in. And so when we look at an equation for photosynthesis, is what we saw was carbon dioxide plus water gives us sugars and oxygen. So what we're going to do is we're going to replace the electrons that we lost from P680 using electrons pulled from water. So what we're going to do is we're going to take the water and we're going to split it. And this is the only place I know of in biology where this splitting of water can occur and the electrons can get given to one molecule to release some oxygen. So what's happening there is the water gets split in a process called hydrolysis, which we've talked about a little bit before, and the electrons get given to P680. So P680 is now being reduced. Water has just been oxidized. That leaves us with some protons, which we'll talk about a little bit later, and the oxygen gas. So that's where oxygen comes from in the general equation for photosynthesis, which is carbon dioxide plus water gives you glucose or sugars and oxygen. So that's really all we need to worry about to start with. So what we'll look at next time is what happens to these electrons, because what we've done here is we've basically taken low energy electrons from water and we've excited them and given them to the primary electron acceptor that I just drew a box around. Now, next time we'll talk about the fate of those electrons. Where do they go? How do we actually get the correct form of chemical potential energy that we need to run the Calvin cycle, which is ATP and a molecule called NADPH. So we'll finish up here with some questions. I'm going to let you work through these, and so I will stay quiet, let you read them, and then I'll flash the answers up. Okay, here's the first question. Let's see how you do. Okay, so we go on to the second question. I'll just add in here that it's okay for you to sit with your book open and go back and work these questions out. So here's the second one. Still talking about reduction and oxidation during photosynthesis. How do you do with this one? Okay, and here's the next question. Let's start trying to tie this to our discussion of free energy that we had a couple of weeks ago. Okay, and here's our last question. Okay, so there's some hard questions in there. You're going to have to go back and look at your discussion, the discussion we had on free energy changes, and maybe a revisit to the material on entropy would be helpful as well, because we can tie this into entropy. So we expect to see some questions on that and some quizzes. We'll wrap it up there, and I will see you next time when we're going to talk about the fate of the electrons that have been energized in photosystems.